Good morning, students. Welcome back to biology class, chapter 13, Photosynthesis in higher plants. Part 2. In the light reaction which is taking place in the presence of light. Light reaction or the photochemical phase includes light absorption, splitting of water, evolution of oxygen and formation of high energy compound like ATP and NADPH. It involves several complexes and the pigments of the complex are organized into two discrete photochemical light harvesting complexes LHC, that is the photosystem 1 PS1 and photosystem 2 PS2. Light harvesting complexes. The light harvesting complexes are made up of hundreds of pigment molecules bound to protein within the photosystem 1 PS1 and photosystem 2 PS2. Each photosystem has all the pigments bound to protein except one molecule of chlorophyll A, forming a light harvesting system that is called antennae. Pigments absorb different wavelength of light for efficient photosynthesis. The reaction center, chlorophyll A, is different in both the photosystems. Photosystem 1, chlorophyll A has an absorption peak at 700 nanometer, that is called P700. Photosystem 2, PS2, chlorophyll A has absorption peak at 680 nanometer, P680. Check one more time. Light harvesting system. The accessory pigments and reaction center, chlorophyll A, together form photosystem PS or light harvesting complexes, that is LHC. There are two types of photosystem, PS1 and PS2. That is the photosystem 1 and photosystem 2. In PS1, chlorophyll A has absorption peak at 700 nanometer, hence it is called P700. In PS2, chlorophyll A has absorption maximum at 680 nanometer and is called P680. These are named in sequence of their discovery and not in sequence in which they function during light reaction. Light reaction or photochemical phase includes first absorption of light, splitting of water, release of oxygen and formation of ATP and NADPH. Process of photosynthesis includes two phases, photochemical phase and biosynthetic phase. Photochemical phase, that is called light reaction. This phase includes, one more time, light absorption, splitting of water, oxygen release and formation of ATP and NADPH. Biosynthetic phase or the dark reaction, it is light independent phase, synthesis of food material. The electron transport system in photosystem center, chlorophyll A absorbs 680 nanometer wavelength of red light, causing electrons to become excited and release 2 electrons from the atomic nucleus. These electrons are accepted by primary electron acceptor, that is called ferredoxin. The electron from the ferredoxin pass to electron transport system consisting cytochromes. The electron moved in downhill in terms of redox potential by oxidation reduction reactions. Finally, the electron reached photosystem 1. The electrons are not used up as they pass through the electron transport chain, but are passed on to the pigments of photosystem PS1. Now, electrons in the reaction center of PS1 are also excited when they receive red light of wavelength 700 nanometer and are transferred to another acceptor molecule that has a greater redox potential. Electron hole created in photosystem 1 is filled up by the electron from PS2. Electron from PS1 pass downhill and reduce NADP into NADPH plus H plus. Now we will see the splitting of water. Electron required for electron transport system is fulfilled by splitting of water, that is called photolysis, associated with the photosystem 2. Photosystem 2 loses electrons continuously filled up by electrons released due to photolysis of water. Water is split into H plus, oxygen and electrons in presence of light, manganese and chlorine. This is also create oxygen as a byproduct of photosynthesis. Photolysis takes place in the vicinity of the photosystem 2. Splitting of water or photolysis, the electrons that are moved from PS2 are replaced by electrons available due to splitting of water. Check the formula. The minerals manganese, calcium and chlorine play important role in photolysis of water. Water splitting complex is associated with the photosystem 2. It is located on inner side of thylakoid membrane. Now we will see the electron transport chain or photophosphorylation. Light-driven reactions of photosynthesis are referred to as electron transport chain. It was first formulated by Robert Hill in 1939. The process of ATP formation from ADP in presence of light in chloroplast is called photophosphorylation. It includes following steps: non-cyclic photophosphorylation, splitting of water, cyclic photophosphorylation. First we will go for the splitting of water or the photolysis that we already discussed. Splitting of water. Now we will go for the cyclic photophosphorylation. When only pigment system or the photosystem 1 is functional, electron is circulated within photosystem and phosphorylation occurs due to cyclic flow of electrons. The excited electron does not pass on to NADP but is cycled back to PS1 through electron transport chain. The cyclic flow hence results only in synthesis of ATP but not of NADPH. Cyclic phosphorylation occurs in stromal thylakoids as these lack PS2 and NADP reductase enzyme. Granule thylakoids have both PS2 and NADP reductase enzyme. Thus, these lamellae conduct non-cyclic phosphorylation. In bright sunlight with a large component of blue light, PS2 absorbs more light than PS1. Hence, both the photosystems work resulting in non-cyclic phosphorylation. In less intense light with more red in the shade, PS1 absorbs more light than PS2, resulting in cyclic phosphorylation. Cyclic photophosphorylation, only PS1 works. The electron circulates within the photosystem. It happens in the stroma lamellae. Possible location because in this region PS2 and NADP reductase enzyme are absent. Hence, only ATP molecules are synthesized. Now we'll see the non-cyclic photophosphorylation. Two photosystems work in series, first PS2 and then PS1. These two photosystems are connected through electron transport chain like Z scheme. ATP and NADPH2 are synthesized by this process. Now we'll see the photosystem 1 and photosystem 2. What are differences? Photosystem 1 located in the grana thylakoids as well as the stromal thylakoids. Photosystem 2 present only in grana thylakoids. Photosynthesis P700 PS1. PS2 is P680, receives electrons from PS2 and receives electrons from photolysis of water. Photosystem 1 performs cyclic photophosphorylation independently. Photosystem 2 performs non-cyclic photophosphorylation in conjunction with the PS1, and the photosystem 1, it hands over its electrons to NADP in non-cyclic phosphorylation, and photosystem 2, it hands over its electrons to PS1. Now we'll see what are the differences between cyclic and non-cyclic photophosphorylation. Cyclic photophosphorylation, what are the characters? Performed by PS1 independently. External source of electrons is not required. Same electrons are recycled. Synthesizes only ATP. Operates under low light intensity. Occurs in stromal thylakoid. Non-cyclic photophosphorylation performed by PS1 and PS2. Electrons are supplied by photolysis of water. Synthesizes ATP and NADPH. Operates under optimum light, occurs in granule thylakoids. The cyclic phosphorylation occurs in stromal thylakoids. These lack PS2 and NADP reductase enzyme. Granule thylakoids have both PS2 and NADP reductase enzyme. Thus, these lamellae conduct non-cyclic phosphorylation. In bright sunlight with a large component of blue light, PS2 absorbs more light than PS1. Hence, both the photosystems work, resulting in non-cyclic phosphorylation. In less intense light with more red in the shade, PS1 absorbs more light than PS2, resulting in cyclic phosphorylation. Thank you.